day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. So far we have discussed the next generation sequencing technologies method, and then we discussed some of the key terms that are important for the course. We will be using those terms throughout the course. We have introduced coverage and quality score, and finally we talked a little bit about experimental design. So, if you are planning to do next generation sequencing experiments yourselves, you will know what are the parameters, what are the aspects that you need to look at. In today's class, we will be focusing on data formats. Okay. So, you have designed your experiment and you have sent your sample for sequencing. You get the data back from the sequencer. So, how would you interpret the data? How would the data look like, right? So, this is something that we will be discussing today, and specifically we will be talking about two data formats in this class. One is the FASTA format and the second one is FASTQ format. Okay. So, these are the two formats that we will be discussing first, and then there are other data formats that we will be discussing in the next class. So, these are the keywords that we will come across during today's class: FASTA, quality score and FASTQ. Okay. So, to give you an overview of the full process, right. So, here is what you have done, right. Up to this point, hopefully you have decided on the sequencer, the platform, the read length that you want to use for your experiment, and you get the read data from sequencer. Okay. And these data can come in different formats depending on the platform that you have chosen. Now, once you have this, right, then the first step will be the data quality check, and this is something we have briefly mentioned. Then there will be different sorts of problems, right. They separate out into two branches. One is the read mapping against the reference sequence if you have a reference sequence already, and this is something that we do if we are interested in looking at signal nucleotide polymorphisms, indels or structural variations, or if you want to do transcriptome analysis, right. We are looking at gene expression patterns or gene expression changes between different samples, and also for epigenomic studies if you are looking at DNA modifications genome wide. We will look into all these different types of analysis step by step in subsequent classes. On the other hand, we have the read assembly problem where we are building a new reference genome. So, this will require different sets of tools, different sets of algorithms, and again we will be discussing this part towards the end of this course. So, here we are now, and step by step we will go along this flow chart. Okay. As you will see, like as we progress, we will come down along this program. Okay. So, today we will talk about these data formats as I mentioned. So, the first one is FASTA format. Okay. So, many of you might be familiar with the FASTA format, what it is. This is a standard sequence data format. So, we use it for storing DNA data as well as protein data. You probably have seen, but I want to just make sure that everyone is on the same page. So, in FASTA format, the header, the first line, it starts with greater than sign. Okay. So, this is something you will see, you will notice, and we will see an example in the next slide. This greater than sign is followed by a sequence ID, which is usually you have maximum of 25 characters. So, it could be anything that you want to put there, sequence that will identify your sequence. And so, these first two parts, these are mandatory, right. These two things would have to be there. The next parts, right, this organism information, the additional information about strain, chromosome, cell line, etc., or if you want to put something like gene information, etc., those could be there. Okay. So, these are optional part, right. This part is optional, and in some cases you will find this information, but you will have this part always, right, this header line with the greater than sign followed by the sequence ID. Okay. So, just to give you an example, this is the sequence of part of chromosome 1 of this species Saccharomyces cerevisiae. As you can see, this is the header line here, right. You have the greater than sign. Yeah, you can see this greater than sign, and this is the sequence ID, right, this ref NC something. Then you have some optional information, right. You have this organism name Saccharomyces cerevisiae, this is the strain, molecule type, this is genomic DNA, right. We are looking at genomic DNA sequence. Then also you have chromosome equals to 1, right. So, chromosome 1, part of chromosome 1. Okay. So, this is the header line, and then you have the sequence information, right. So, the actual sequence information you can see this CC, SE, etc., right. So, this should be your FASTA format. You have probably seen this before. So, Sanger sequencing data, it turns out, so get two files. Okay. So, one is called trace file. For example, you will see something like sequence 1.ab1, right, just as an example. So, these are called AB1 files. You will, whenever you do Sanger sequencing, you will get these AB1 files. How do they look like? We will see in a moment. Okay. So, it is important to actually see how Sanger sequencing data looks like, and then we will compare against the, so compare with the NGS data sets, right. So, this is something what you get in Sanger sequencing data, the trace file. In addition, you will get this sequence in FASTA format. Okay. So, we just talked about FASTA format. So, you will get this sequence 1.FASTA or sequence 1.FSA, something like that. Okay. Now, if you open this AB1 file, right, you will see something like this. So, this is called the chromatogram data, where you have this sequencing data stored. Okay. So, what you see here is, so you have like 3 rows of information, 3 rows of sequence here in this diagram. So, the first row, this is the sequence, okay. This is the sequence in the first row, okay. And for each base you have a peak, okay, and these peaks are also colored differently, right. So, if you go back to the Sanger sequencing principle, how it works, we have these different DDNTPs, right, they are labeled differently. So, these labels are kind of shown in different colors here, right. So, T is in red, right, C is in green, C is in blue, A is in green, and G is in white. Okay. So, 4 different colors you see here. Okay. And for each of these base you have these peaks, corresponding signal peaks, right. So, for some it is very high, the peak height is high. So, the signal intensity is high, whereas for some the signal intensity is slightly lower, right. So, but these signal peaks are well resolved, right. So, you can call these bases one by one. What you will notice is this first part here, right, is that the peaks are almost non-existent. Okay. You do not see these peaks, right, for the first 10, 12 bases, these peaks are not there. Okay. Although there are bases called, okay, but you will probably can guess that these bases will have low quality score, okay, because the signal intensity is lower and quality score would probably reflect the signal to noise ratio, right. Okay. So, let us look at the quality score, right. So, we can actually also look at this quality score of this sequence. For each base you will have a quality score, we have discussed this, right, how quality scores are assigned. So, here is the quality score, right, for this base, for example, right, and the height of this column actually represents the quality score. So, higher the column, the better the quality. Okay. So, as we have mentioned, like in the beginning the quality score is low. So, you see this column height is very low, right. So, you have low quality. So, this is something that is common in Sanger sequencing, and well, this is a well known issue. Okay. So, as I pointed out, this is the low quality value at the start, right. In addition, if you sequence, let us say 1 kb DNA, 1000 base pair DNA, right, you will start to see quality degradation at the end as well. Okay. So, here you can see the number, right. So, we have already sequenced about 1000 bases here, right. So, you see this number 1000 here. So, we have already sequenced 1000 bases here, and as we go along, right, now the quality starts to drop, right, starts to fluctuate. So, before this 1000 base position, like the quality was more or less okay. Now, the quality starts to fluctuate a bit, right. You can see this here in this row, and then it goes down further, right, here, and you can also see the peaks, right. These are not so well resolved anymore. Okay. There seems to be some overlap between the peaks. Okay. And that is probably the reason why this quality score goes down, because you are again measuring the signal to noise ratio. So, if there is a lot of noise compared to the signal, you will get lower quality score. Okay. So, these are two well known issues we know. So, the question is, why are the quality scores low in the beginning? So, one is in the beginning part, if you remember the Sanger sequencing principle, we have these DDNTPs, right, and we are generating fragments of all sizes. Okay. Now, for very small fragments, the electrophoretic medium that we are using in capillary electrophoresis is not able to resolve those fragments very well. Okay, these are very small. So you can imagine these are like a DNA agarose gel or a protein gel, right? You have some resolution power. Now this electrophoretic medium, right, this cannot resolve very small fragments very well. Okay. This is one reason. The other reason, of course, you have this DD-NTP addition, right? These also have some these also have higher molecular weight compared to normal DNTP, right? And they will shift the small DNA fragments much more than they do for the bigger fragments. Okay. So, this will kind of change the positions, expected positions of these small DNA fragments in the electrophoretic medium. Okay. So, these two factors contribute to this low quality score. Okay. So, the detection does not work that well. What about the low quality score at the end? Okay. So, this is something we have seen. Again the resolution power comes into play, right. So, this electrophoretic medium, it has some optimal range, right. So, probably around 50 to 1 kb, right. So, 50 base pair to 1 kb length, right. Beyond that, it's the resolution is not very good. Okay. So, again what happens is this fragments, they tend to now start overlap with each other, right. So, they tend to overlap with each other, and which means you get this overlap in the signal peaks. Okay. And that we have discussed, right. Now, we are zooming into that, and you can probably notice some of these overlaps here, right. For example, here you can see the signal is for this T, right, in red, but there is also a background noise in gray, which is for G, right. So, this background noise is almost equal, right, in magnitude to the signal. Okay. So, that is why the quality score, if you notice, is substantially lower here, right. So, this is kind of the situation you will probably see in many places now. Okay. The quality score going down because you have this signal to noise ratio going down. Okay. And probably you can see here, for example, or maybe here also, right. You can see, you can notice these events if you look at this chromatogram data carefully, you will see this lower signal to noise ratio. Okay. So, that kind of gives you an introduction to the sequencing data format for Sanger sequencing. Okay. So, now we can move into Roche 454 data format, right. So, this introduction would be helpful as we will see for 454 data format, because the sequence data that you get from 454 is stored in FASTA format. Okay. So, in 454 you get one standard flogram format file, so which you will see like sequence 1 dot SFF or star dot SFF. So, star is the name, right. You can give any name that you want, and then this is equivalent to the AB1 file that we have just seen. Okay. Right. So, we have seen this, this information about sequencing is stored in the AB1 file. Similarly, in Roche 454 this SFF file, it actually stores this information, sequence information, right, what actually happened during the sequencing process. You can read this file and you can generate two different files. Okay. This is something that is done by the program, right. When you get the output from 454 data, you will get these two different files. You will get the SFF file, along with you will get this sequence file, which is the sequence 1 dot FNA, for example, and you will also get a quality score file, which is the sequence 1 dot qual. Okay. So, you will see these two files typically. Now, the sequence file is actually stored in FASTA format. Okay. So, which means we have learned how the FASTA data would look like, FASTA format would look like, right. So, here is the header, for example, you have this first sequence here. So, this is the header, and you have the sequence ID, right. This is the sequence identifier, and then you have some extra fields, right. These are optional fields that are given in the 454 run. Then you have the sequence, right, the actual DNA sequence here, and you have a second header line. Okay. So, this is how the data is stored. You have first header line, you have sequence, then you have another header line, the sequence, next sequence, right, and this goes on, right. So, if you are getting 100,000 read data from 454, you will have these 100,000 sequences one after another. Okay. So, this will be quite a big file. Okay. You can imagine how much data will be there. Okay. So, how do you open that file? How do you look at that file without crashing your system? We will talk about that when we discuss the hands-on tutorial. Okay. Now, what about these other fields that you have there? Okay. So, these are the fields that we see. So, as I said, this is sequence ID actually refers to the read ID, right. So, this is the read number that we are getting. We have the read length, right. So, here is 673 bases here. Then the xy is the position in the picotiter plate, and xy and region, this gives you the position in the picotiter plate. If you remember these picotiter plates where you have these wells, each well contains one bead, right, and on the bead you have these DNA fragments. Okay. So, this gives you the position in the picotiter plate. Okay. And then followed by that you have this run number or experiment number, right. So, probably you can see the date and that probably the time when this experiment was run, right, and this kind of information is there. Okay. So, then you have, so once you have the sequence file, you also have the quality score, right. So, in SandGuard you get the quality score in AB1 file, right, and since you are looking at one sequence, you can probably manually go and check the quality score for each of them, right, and maybe you are handling maybe 8-10 sequences at a time in parallel, and that is manageable. In 454 you are dealing with 100,000 reads, right, 100,000 sequences at once. So, you need to put quality score in some file where you can process them by writing a program, right, or some software tools can process this quality score. Okay. So, this quality score is stored in a separate file, as I mentioned, is the .qual file. Okay. And this stores the quality scores in FASTA format and in the same order as in the sequence data. Okay. So, if you have read XYZ in the sequence file, right, the order in the qual file would be the same XYZ. Okay. So, that you can map, right, if you are just using a program, you can map these very easily back and forth. The read, you can read the sequence and you can get the quality score. Okay. So, this is how the quality score files will look like. The header line is here, right. This is again in FASTA format. You can see the header lines for those same two read data, right. So, this is the read ID here, then you have the length, etc. The same information is there, exactly same information, but the difference is now you see the numbers. Okay. Instead of this ATGC, here you see the numbers, right. For example, 21, 21, etc. So, each number is a quality score for a base in the sequence data. Okay. So, the first number is the quality score for the first base in the sequence data, second number is for the second one, so on. Okay. So, this in this way you have this quality score for all of them. Okay. And then again you follow the FASTA format. So, you have, so once you have mentioned all the quality scores for one read, then you have the header line, then you have the quality score for the next read. Okay. So, this is how the whole thing is stored. Okay. So, this is just to highlight, this is a quality score of one base. Now, what are the drawbacks of Roche 454 data format, right. So, it looks great, right. You can now look at the sequence data, you can read the quality score, right, and you can check the quality score of the bases, etc. You can write your own program, or you can probably use some software tools that will read the quality score as well as the sequence data and will give you an idea about the sequencing run. Okay. So, this is important, the quality score is important as I have shown you in the flow chart, because the next step is the quality check, right, or quality control. Okay. So, for that we need this quality scoring there. Okay. We cannot just look at the sequence data and say, okay, this is of great quality, right. We need to have the quality scores alongside. Okay. So, what is the drawback, right? We have the quality score in 454 data. So, what is the drawback? Why do not we use this FASTA file and quality file separately, and then we have the data? So, one thing you probably would notice is that the qual file, the quality file actually takes more storage space, okay, more than 2 times the space of the FASTA sequence file. Okay. If you go back, right, this sequence file is ATGC, there is no space in between them, right, just one letter at a time you are storing the data. Here in the quality score file you have the numbers, right, you have this two digit numbers, right, is 12, usually the quality score is between 0 to 40, right, and maybe it is 12, 15 most of the time. If it is less than 10, it is actually very low quality, right, it is probably not good. So, most of the time it should be above 20, 15, 20 at least, right. So, you have these 2 digit numbers for every single base in the sequence, right. So, you are generating these 2 digits for every base that you have. On top of that, you have the space between these numbers. Okay. So, that takes up a lot of storage space in the hard drive, right. So, if you are generating a lot of this sequencing data, you will probably run out of space very quickly if you have this kind of system, right. You have this quality score and these numbers stored by spaces, right, differentiated by spaces, right. So, this is something that is not very efficient, right. The second problem is finding quality scores of individual bases is computationally time consuming. Okay. Why? So, if you can you can think of, right, in this case, right, you have this read 1 and you have the quality score. You can write a program that will say, okay, read 1, find this read and look at the quality score of these bases. Now, this is okay for maybe first few bases or maybe first 100, 200, 1000 bases reads, right. What about the reads that come at the end of this file, right. So, if there is a read that is like, let us say in the 10,000 position, right. So, you have to, once you are reading this file, in the other file you will also first read all the 9,999 reads, only then you reach that 10,000 reads, right. So, it is a very inefficient process. It will take a lot of time, because every time you are reading one read sequence, you have to read like the quality score, you have to traverse through that file. Okay. So, this traversing through the quality file, it actually is very time consuming. Okay. And this is especially hard for the reads that appear in the middle or at the end of this sequence file. Okay. For the first few reads, first 1000, 2000, 5000, maybe you will not notice the time taken. Okay. But as you go along, right, as you reach towards the end of this file, getting quality score for every base in the read will take much longer than it used to take, right, for the first or second read in the file. Okay. So, this is something that is not very efficient. Okay. So, here is an example, right. So, for example, you see here, this is the sequence file. Okay. This is the in FASTA format, where you have the read data and probably you have reached about the 99%. So, here is the like towards the end of that file, right. So, here it is in actually in Unix terminal, right. So, it shows you the percentage of file that you have already covered. Okay. So, it is we have covered already 99% of the file. This is the line number that we are at, right, and this is about 300,000, right, 335,000. Okay. So, this is the sequence. Imagine we are reading here somewhere here. Okay. In the other quality file, we also have to read these 335,000 lines before we reach the quality score value, right. So, you can imagine this is going to take a lot of time. Okay. So, how can we address these drawbacks, right. So, this is something that we saw in 454, right, that we have this quality score and the sequence data separately, and this is computationally really inefficient, right. So, how do we address these drawbacks? We cannot just throw away the quality score, we need that for the quality control in the next step. Okay. So, there could be, you can think of some solution, but probably the best solution is, why not put the quality score along with the sequence data in the same file, right. And if you can find some way, right, to put the quality score along with the sequence data, right, very close by, that will serve a purpose. Okay. Because then you do not have to read two different files, you do not have to traverse through all those lines. So, the moment you are reading the sequence data, you are also reading the quality file. Okay. So, which means you require a new data format, right. It is not going to happen with FASTA format, you are going to need a new data format, right, and that is something that is required. So, what you need is a data format where the quality scores are stored in a more efficient manner, right. So, one is you are storing these digits, right. So, 2 digits for every base, then you have the spaces between these numbers, right. Without spaces you cannot distinguish, right, which quality score is for which base. Okay. So, this again we probably can do better, right, we can have more efficiency there. So, if you can do that, right, this will require less storage space on the computer, right, or in the server. So, this is something that probably is desired, right. So, we need a data format where the quality scores can be stored in more efficient manner, and also where the data quality scores are stored alongside the sequence data, right. So, these two requirements if we can fulfill, that will be of great help for us. Okay. And it turns out that there is this data format that has been defined now, where you can do this. So, it is called actually FASTQ format. Okay. The name comes from FASTA format with quality score. So, you have this Q means quality, right. It still it resembles the FASTA format to some extent, right, but it also contains the quality score in the same file. Okay. In addition, right, so you have this sequence and the quality score in one place in the file. In addition, you have the quality score encoding, right. So, these quality scores are not represented by numbers anymore, those like inefficient two digit numbers, right, and no spaces. So, you have more efficient way of doing this through some encoding, some sort of encoding. Okay. So, just to give you an example, so this FASTQ format is used by Illumina platform, right. So, all Illumina data comes in FASTQ format. Okay. And just to give you an example, this is the FASTQ data format, right. So, you will see this looks like FASTA, but not exactly FASTA. Okay. So, one of the things you probably notice, again you have these header lines. So, this is your header line, the information, what is this information that you have in this header line, we will discuss in the next class. Okay. There is a lot of information here, and what you will probably notice is this header line starts with this @ sign. Okay. So, this is a difference with the FASTA file. Then in the next line you have the actual DNA sequence, right. You see this CSCC, TTA, etc. right. This is the actual DNA sequence. Then you have a plus sign. Okay. This is actually a spacing, right, between the sequence and the next line. So, what is the next line? So, this next line actually contains the quality score, okay, and this is actually an encoded quality score. Okay. This you do not see the numbers anymore, it is not 21 or 25 or 35, right. You see some letters, some symbols, right. So, there is an encoding that is used, and this makes it much more efficient, right, in terms of storage space. You do not have any more space. So, it takes exactly the same amount of space as the sequence itself. Okay. So, this is something that we will see, we will discuss again how this encoding is done in the next class, right. And plus is the spacing between the sequence data and the quality score, right. So, this is something you probably now see, this is a much more efficient way of storing this next generation sequencing data, right. You have the sequence along with the quality score, right, in the same place. So, when you are reading this file, for every read with this identifier, right. So, here you have this identifier here, right. For every read you have the sequence data along with the quality score, right, just one after another. And if you are using a program, if you are writing a program yourself, you can just go through this very quickly, and also the data is stored in a very efficient manner because it is not taking too much space. And this kind of repeats, right. You have this one, then you have this next read, here is the next read, right, and for each read you have the sequence information and the quality score. Okay. So, what you will see, like most of these files, right, they come in this sequence .fastq format, right. So, the name will be some sequence name, some name .fastq, and most of the time they are also stored with this .gz compression. So, this is a compression in Unix, and this also again reduces the file size. So, the final file name would be sequence.fastq.gz, right. So, this will be useful when we actually do the hands-on tutorial. Okay. So, these are the references that we have used in this class, and to conclude, so Sanger sequencing data is stored in FASTA format. So, that this is something what we have seen, and sequence data from 454 platform that come in FASTA files, and quality scores are provided in qual files, and separate sequence and quality files are inefficient for computational processing. Probably discussed why this is so, and this drawback is addressed by the FASTQ format, which combines sequence data and quality score in a single file. In the next class, we will be talking about the FASTQ format in much more detail, what kind of information it stores, and we will be looking at actual sequencing data. Thank you.